Good morning, everybody. Welcome to the webinar. We are going to get started in a bit after everybody gets a chance to join. I think if you are one of the webinar speakers, I'm trying to promote you to panelist. You should be, yeah, there you go. So is the Sciensano room, is that Marie? And yeah, is that you guys? Okay, great. So you guys will go first if that's okay. I'm sorry that you didn't get on the panelist link before we started the webinar, but can you guys hear me?
Yes, we hear you.
Okay, great.
So we'll try to share the presentation.
I have some intro slides to share first. And then
Okay,
we'll
I will
get started.
unshare.
Yeah.
Stop. Okay.
Great. Okay, I will share the intro slides here. And I'll go ahead and start going through them. We'll start in the beginning. Okay, welcome to the Wastewater Surveillance SARS-CoV-2 and Emerging Threats continuing webinar series that Kyle, Bibby, and I have run with the assistance from Sarah Philo, who is here today co-hosting. If you are not on our mailing list, please scan this QR code. And if you have nominations for speakers, also please scan the QR code and send us ideas, or just email me is also fine, or Kyle. Today, we're very excited to have two sessions with three speakers. So the first, we're going to have Nancy Roosens and Marie-Alice Fraiture, and I hope I'm saying it like close to right, from Brussels. And they will be talking about differentiating between livestock and human sewer inputs. Very excited to hear about their work. And then also very, very excited to hear from Shannon Metzinger from Colorado Department of Public Health, who's had success sequencing influenza from wastewater, and really excited to hear her talk as well. Just a plug for May's speakers. We have two junior scientists, an assistant professor and researcher at Harvard, Catherine Hoare and Hannah Healy. We hope that you guys will join to hear about their work as well. Okay, so with no further ado, we will have Nancy and Marie-Alice share their screen. And I'll stop sharing and then we'll get started. So remember, for you, everyone in the audience, if you have questions, please put them in the Q&A, which if you don't see it at the bottom of your screen, you would hit those three horizontal dots to find it. And we'll ask questions at the end of the 20-minute webinar. Okay, I will be quiet now and pass it off. Thank you.
Yes, so I will start with a very short presentation of Sciensano. It will take 1 or 2 minutes. And then Marie-Alice will explain the context. And then Marie-Alice will continue with the scientific topic. So just to mention that Sciensano is a Pasteur Institute of Belgium. And so now we are a One Health Institute because there are several fusions. So now we are incorporating human health and animal health. And also we pay a lot of attention to the environmental pillar. And it's why wastewater is very important for us. So just to mention that Sciensano is also with 1,000 people and 750 scientists. And our mission is to integrate research, data, and scientific expertise in order to advance in One Health. And this, of course, in collaboration with partners in Belgium and outside. And it was why we were very happy that you invite us. So thank you for that. So we are working through 3 pillars: scientific service, scientific expertise, and scientific research. So here you have an overview of all the organization of our institute, and we are there in this department, biological health risk, and we are a transversal service, and our name is transversal activity in applied genomics. And for wastewater, we are working in close collaboration with two other services, epidemiology of infectious disease and foodborne pathogen. Regarding our service, so the goal is to drive innovation within Sciensano, and this through a genomic method. So we are doing research, expertise, and service provision. This is the organigram of the service, transversal activity in applied genomics. We are approximately 40. And we have one very specific service, GMO Lab, who is working on GMO, and two other services, the Biotech Lab and the Bioinformatic Platform. And there it is transversal service. And we are working in developing solutions for everything related to high throughput sequencing, genomic, digital PCR assay, and also to deal with the massive amount of data using a centralized bioinformatic platform. So approximately half of the staff are bioinformaticians. And Marie-Alice is there, is a senior researcher, and she make the link between the three units, in fact. Regarding the service provision, I will be very short, but we are doing with all the type of sequencing. Of course, we are offering scientific support for this. We are working, we have core facilities that we are offering to the whole institute. And also we have a lab, a GMO lab, we specialize in everything related to GMO detection. We are also, we have a lot of, we provide expertise in applied genomics at EU level and also at Belgium level. So everything related to GMO and also high throughput sequencing and bioinformatics. And there it's a list of our research topics that are often transversal. And yes, one of the big pillars of our research is genomic surveillance. And in this context, we are developing tools. And yes, it's for this that we are one of the three labs in Sciensano working on wastewater, because we are developing everything related to improve the surveillance of wastewater in Belgium. And it is in this context that Marie-Alice and Laura are working. Marie-Alice is more working related to everything related to AMR, although Laura is more working on everything related to virus. And also we are developing, because I see one of your next step, sequencing for influenza, wastewater, another type of virus. But this we could maybe discuss another day. Voilà, I think I have finished.
Yes.
For this part. Now we go in the topic.
Yes. Yes. So I will now speak about the reason why you kindly invite us. So our latest study about enhancing One Health thought through wastewater-based surveillance, development and validation of a multiplex ddPCR tool to differentiate human and livestock contribution. So as you all well know, wastewater surveillance is a useful tool for public health monitoring. It has been well demonstrated a few years ago with the SARS-CoV surveillance, and more recently with the antimicrobial resistance gene surveillance. However, one thing with this wastewater surveillance is that it's mainly focused on the human population, but we didn't have any idea about the livestock contribution to this wastewater, and so it's something that we wanted to explore. So the knowledge gap are that we don't know, in fact, how much do the livestock contribute to the microbial and genetic content in wastewater. This uncertainty affects accurate data normalization and interpretation of surveillance results. And this is especially problematic, I would say, with the antimicrobial resistance field monitoring, because it can come from both human and animal, not like the case of SARS-CoV, that it was clear that it was related to the human population. And this kind of issue is particularly crucial for the One Health context. Yes, so in order to investigate this topic, we had the following roles. So we wanted to develop tools to evaluate whether livestock significantly contribute or not to the wastewater inputs. And also we wanted to improve the normalization and the interpretation of wastewater surveillance data by including animal fecal source. For this, our plan was to develop and validate duplex digital droplet PCR, targeting host-specific fecal indicator marker, both for human and also for key livestock species that are important, that are in fact chicken, cows, and pig. So I guess you are familiar with the development of a method, but I will just give a small word on this, and sorry if it's a bit too much of detail, but I will just keep it very short. So for the development of a PCR and here a digital droplet PCR method, we need to select targets, DNA targets of interest. And for this, we are designing a set of oligonucleotides. So we have here a forward and a reverse primer that will allow to amplify the region of interest of the fecal indicator. And we are, in addition, designing a probe that will angle specifically to the region in order to inform us if the region is there or not. So then after the development, we are going for the validation step. And it includes key steps that are first the evaluation of the specificity of the method. So need to give a signal only with the target sequence and to not give a signal with the non-target sequence. Then after this, we need to assess the sensitivity in order to see what will be the limit of detection of the method. And finally, we are looking for the applicability on real-life sample. So first of all, before to start anything, we needed to find some sequence to target. And where we had a look to the literature, and we have identified already real-time PCR methods that were developed and targeting some fecal indicator related to our target of interest that are here. And so here you can see that according to the hosts, we have different methods that we have listed. The ones that are in green are related to, in fact, the microbiome associated to the hosts. And the one in blue are, in fact, directly specific to the DNA of the host. So from all this list, we have done a first selection, and then we decide to test already this one on a minimal set of samples in order to assess if we can continue with them or not. So when we do a specific validation, in fact, we usually start with the specificity and more particularly a specificity in silico in order to see if according to information about the sequence that we have the expected result or not. However, one issue here is that it's not a classical way to test the method, because normally we just we design a method to amplify a region that is specific to a species, and then we are testing the specificity in silico against sequence of the species and or not of the species. But here we are looking for fecal indicators. So the problem is that the database doesn't contain or didn't contain clearly complete information about the macrobiote of the hosts of interest. And so it's quite, it becomes quite difficult to assess the specificity. We have also looked to the some data sets that were available regarding some available metadata for microbiome. However, we had some difficulty at that time that the sequencing depth was not enough or the annotation was not enough. So, and especially for non-human and less studied animal species, so we could not go properly for this option. So we had to go directly to the second step, that was the experimental part in the lab. And then it also required, this is a novelty here for this kind of approach, is that we had to create an in-house fecal DNA bank by collecting, in fact, fecal sample from different well-identified humans and animals. And like this, each time we had like some fecal sample, for example, from pig, another one from cow, and then each time we extract the DNA from this sample and we create our database. In total, we have a fecal DNA bank of 41 samples, and that cover 14 species. And of course, we had some fecal sample from human, chicken, cow, and pig. From different specimens and different locations, but we also include other species of animals, so other birds and other farm animals, I would say, and also pets. So then, with all this DNA, so we first wanted to test the method. The qPCR method were transferred to digital droplet PCR, and we first tested in simplex. So what was the goal is that we test the method targeting the fecal indicator for the cow with a list of positive and negative expected signal material, and then we observe if we have the expected signal. And in this case, for example, for the fecal indicator for the cow, we had only a positive signal when we have the fecal DNA from a cow and not for anything else. It was the same case for the pig fecal indicator. Then we also test the two methods for human, and we saw that this one for targeting cross-page was not giving expected results. But the one that targeting bacteroides was giving only a positive signal for human and not for the other. Finally, for the chicken, it was a bit more challenging to find a good method, but finally we tested 5 different fecal indicator PCR method, and we find this one that gave only a positive signal when we had fecal DNA material from chicken and not when it was something else. So finally, we find our targets and methods, and then the goal was to make it to combine this simplex method in duplex method. And for this, we made some optimization in silico in order to see if the methods were compatible. And finally, so we had 2 duplex. The first one was combining the method specific for the fecal indicator of human and chicken, and the other one was combining the methods specific to the fecal indicator of cow and pig. We go for the full specificity of these 2 duplex methods against the full in-house fecal DNA bank. And as you can see here, so we had only a positive signal for the expected material each time. So for the here in this duplex, you have the fecal indicator method for the human, and we had the expected result only positive signal when we have the human fecal DNA. Idem for the cow and the pig. And in case of chicken, we also had a very nice signal when it was the fecal DNA, fecal DNA from chicken. But we also observed a very low, very weak signal for one of the three dog, but it was very, very weak, and I will go in more detail now. So for now, very good and expected specificity for the human, cow, and pig fecal indicator. Then we explore a little bit more the chicken method, chicken fecal indicator method, because we observed that for one of the three dogs that we test, one was giving a weak signal, positive signal. So we also had a look in the literature, and this method could be influenced by the if you have a chicken-based diet. So this can be an explanation because, in fact, in contrast to the three other methods, here for the human, the cow, and the pig, this method are targeting bacteria that are inside that are host specific. But here, with the method for the chicken fecal indicator, it targets directly the DNA of the host. So if you are eating some chicken, it's possible that then in the fecal material, there is some trace of the chicken, and then that can give a low signal. However, in order to go further and to demonstrate it, we have done a small feeding experience with a volunteer dog. So for this, we have we had applied the following experimental workflow. So one dog was fed 2 weeks with only fish, and then after 2 weeks with only chicken. And at the end of each week, a fecal sample was collected, and we extract the DNA and we applied the method PCR. And we saw that when we had only salmon in the foods, we didn't have any signal for the fecal indicator for the chicken. But when it was fed with chicken, we observed again this very, very weak signal. So we could demonstrate that he has the chicken diet had really an influence, but of course it's very, very limited because it's super weak. But at least we could explain it. So based on all this information, we could say that the four methods and therefore the two duplex were specific, as no false positive and no false negative were observed. Then after the specificity, as I say, we were looking for the sensitivity evaluation of the methods. And for this, we are taking the targeted sequence for each method, and then we do serial dilution of it in order to have an idea to reach the limit of detection. And usually we are testing the range of 20 copy up to 0.1 copy of the target. Then you can see that with all, so for each method were tested like this, and we got a very, very beautiful sensitivity. So it's super sensitive, and we have below 25 copies. So between 4 and 9 copy, we're able to detect it. Well, it's the limit of detection, and it's completely in line with international standard and guidance for evaluation of PCR methods. So we were really satisfied, and it showed that it's in line to say that it's sensitive. In addition to this symmetric sensitivity, we also wanted to test the asymmetric sensitivity. So what is it? It's not really complicated. It's just that, in fact, for the method, for a duplex method, we are mixing the two targets. So for example, here, for the duplex one that targets the human and the chicken fecal indicator, we will put in the same reaction the target sequence for the human fecal indicator and the target sequence for the chicken, but with an unbalanced way. So very high amount of one with a very low amount of the other. And then vice versa, this one very low and this one very high. And then it's in fact to see if the detection of one, if it's really highly present, can affect the detection of the other one that is really at trace level. But in any case, we observe the expected signal without any difficulty. So we could say that there is no cross-reactivity and we had a very satisfying and expected result. So for this point, again, it was a check and then we could go to the next step. Then we go for the applicability. So for this, we had performed, we have used some raw wastewater, and for which we have tested first with just raw and with all the methods. And we have also spiked this raw wastewater with either some chicken feces or with pig feces or with cow feces. And what we could observe is that in the raw, so the unspiked wastewater, we had a signal for a human fecal indicator, which is quite expected because it's from wastewater treatment plant that was collected from, in fact, the capital of Belgium. So it's like a lot of people, so it's clearly associated to human population. And we had also a small but very weak signal for the chicken, but could be related to the diet effect like we observed with the dog experiment. And then no signal for the fecal indicator of the cow and the pig, which is also normal because there is like no farm, maybe one. So in this
I want to
area,
make sure we have time for at least one or two questions. So hopefully this is your last slide. We're almost
no, I had like against few, but I don't know, or
yeah, because
I think in 5 minutes I can finish.
we don't have 5
but
minutes.
if it's too long,
yes.
Yeah, I thought it.
Sorry. You can do one more slide, I think, but then we'll have time for like one or two.
Yeah, okay. So I will go to the essential. So in fact, so we have tested the applicability by spiking or unspiking some samples to see if we had the expected signal for each time the method, and if in fact it was compatible with wastewater. What is the goal of this approach? And we confirmed that we had the signal expected and that the fact to use wastewater was completely convenient for the approach. And then we also did a pilot monitoring over 1 year on 2 sites. That was one in Brussels, wastewater treatment plant in Brussels, so the capital. So a lot of people and not a lot of animals. And then in an opposite site was also taken where there is, of course, we are in Belgium, so we have a lot of people, but the most abundant farm area, I would say. And then like this, we could see if the method was applicable for such kind of pilot monitoring. And I would just, the key idea was that we compared the signal that we obtained for this fecal indicator here from the city and the city plus farm, I would say, area. And we saw that we have a signal for the human very high when it's in the city, and still the chicken, so related to the fecal, probably related to the diet impacts. We have the similar trends in the area where there is more farm, but then we see also the presence of the fecal indicator for the cow and the pig. So we can see that according to the site, there is really an impact of the indicator. There is really an impact of the livestock contribution. And then, here is just for the diet influence. We saw it. And
can I interrupt you?
and voila.
Yeah.
That's all I think.
Yeah, sorry to cut you off. Very interesting. We only have time for one question, but I'll just say I really like the way that you did all those controls, and I was really impressed with like the accuracy, I guess, of how things were working. And the question, there's some questions in the Q&A which you can also answer offline, but the one question I think that we'll ask is about diet, which is, you showed in that super interesting dog feeding study that you could detect the chicken after the dog ate chicken. And how can we distinguish between diet-based inputs with the I think it was a mitochondrial or genomic marker in chicken and or other animals and actual fecal inputs. How are you thinking about that?
So what do you mean exactly about this?
Like
So
if you eat chicken and then you excrete chicken DNA, how do you determine if that's happening or if there's actually chicken feces entering the sewage? How do you differentiate?
Yeah, in fact, we observed so with the digital droplet PCR, you have the presence, absence, and also a number of copy, absolute number of copy that is calculated. And we observed that, in fact, when we had really fecal DNA, fecal DNA from chicken, the signal was, the number of copy detected was very high. However, when it was just from diet, we had a signal, but at very weak, so very low concentration at trace level.
Yeah, so it means that at low level, we cannot distinguish between a low level chicken.
If there is like one farm that is connected with a big city, we will not see it. But if there is a lot of chicken farm, then we can really see an influence.
So then if we want to be more accurate, what we may maybe do in the context of avian influenza and all these things, there we have to develop a fecal indicator based on microbiota.
Yeah, it was really a tricky thing because it was our main idea first to have, like with the other fecal indicator, to have like bacteroides, something like this that go to target the bacteria inside the fecal material, but all of them were not satisfying regarding the specificity, and so we have some false positive and false negative. So it was not acceptable, but it's true that the ideal will be anyway to have a bacteria, but apparently the method that were existing, we didn't have a bacteria or a group of bacteria enoughly host specific. So it was.
Yeah, thank you so much. I know there's a lot more questions. Unfortunately, we're out of time, but you can answer some of the Q&A if you'd like during Shannon's presentation. So thank you so much for that important work and great presentation. And we're going to move on to Shannon and Sarah will moderate Shannon. Thank you.
Great. Thank you guys so much for inviting me. As mentioned, I'm Shannon Metzinger. I'm from the Colorado Department of Public Health and Environment, where I run our genomic surveillance program, which includes wastewater detection efforts for the state of Colorado, and as part of our Center of Excellence activities through the CDC's National Wastewater Surveillance System. We also this group also is our sequencing core and performs most of our sequencing efforts, both for wastewater and clinical efforts at the public health level, and our bioinformatics team. And so today I'll be talking about our efforts to develop and implement seasonal influenza wastewater sequencing efforts and how that has been turning out in real time in Colorado. So they move forward. So these are some resources. We've got a preprint on the tiled amplicon approaches I'll be talking about today. On the right, there's a link to our GitHub with our bed files and all the pieces needed for the sequencing approach. And then on the bottom, the data linked to the recent preprint, which doesn't include some data I'll be talking about now, which goes a bit beyond what we've been able to put up as a preprint, but is under consideration for publication now. So when we started thinking about sequencing influenza from wastewater, obviously wastewater is a complex matrix, but influenza is also a complex virus. There's a lot of genetic diversity, obviously, between this pathogen, even at the seasonal level, right? H1N1, H3N2, and influenza B viruses as well that circulate seasonally. Another piece for wastewater is that makes it complicated is that influenza has a segmented genome. So obviously all those different gene segments are not linearly connected like SARS-CoV-2, where you could assess an entire virus by tallying across that entire genome and connecting it that way. If we want to look, we don't know what HA, if we sequence it in wastewater, is connected to which NA. And so you could get, an H5N1 and you don't know that's coming from seasonal H1N1 and H5 with another NA attached, right? So that makes it a bit complicated. And then finally, obviously, when we're talking about wastewater, we're talking about fragmented genomes. And so being able to identify those really fragile RNA viruses very sensitively, because obviously SARS-CoV-2 was shedding at the peak of the pandemic at a higher level than we're seeing for a lot of respiratory viruses, including influenza and wastewater. And so how do we sequence this very sensitively? And so these are a lot of questions we asked ourselves when trying to develop an approach. And then, of course, when we talk about sequencing in wastewater and we talk about sequencing, there's a lot of different ways that we could be doing this in wastewater that a lot of different groups are exploring right now, including agnostic approaches, so sequencing metagenomics, straight shotgun sequencing from wastewater without actually needing to know what you're looking for when you're doing that sequencing, or a broadly enriched approach like hypercapture for a large panel of different pathogens, where that could be way more sensitive for the pathogens they're looking for, but you could span quite a large group of different pathogens that's a bit more sensitive than agnostic approaches. And then, of course, your targeted or tiled amplicon or amplicon approaches, where you need to know exactly what you're looking for. You need to design it very close to what you're looking for. But you may get a better depth of understanding of diversity within that small piece or pieces of the genome that you're looking at. And so there's different pros and cons to all of the approaches. In the case of our efforts for influenza, we decided to focus on a tiled amplicon approach, specifically for the HA gene, basically to start with that minimal piece of information from flu that can generate a lot of impactful information, right? Because the HA gene is both highly divergent and necessary for immunogenicity or evading immunogenicity or immune systems. So I think Michael, who's the first author on this Oliver paper we're referencing and utilized to make our approach, I think he presented maybe last time on this webinar. And we utilized the Oliver tool that they developed at Rice with Lauren Stadler's group and Trot Hagen's group. We like to utilize this approach because, one, we can make pretty small amplicons, which we thought would be useful for increasing the sensitivity of what we're looking for. In addition to that, we could perform a diversity study of influenza viruses, utilize that the variant information or VCF file to feed into this primer development or scheme software or algorithm to generate primers that focus on areas that are most divergent wherever we can, or most conserved within the HA gene wherever we can. And so we pulled down over 4,000 H1A or H1 HA gene segments from public repositories, over 7,000 H3 HA genes, and over 2,000 B Victoria gene segments from public repositories that were collected between 2018 and 2023 when we started this effort to perform this diversity study and utilize it for developing a primer scheme. We've ran these, the diversity that we saw from from each gene segment, H1, H3, or BVIC separately, to design a primer scheme for the HA gene of each of those seasonal HA genes specifically and separately because of the high diversity within influenza that I mentioned. And we focused on making a 125 to 250 base pair average amplicon scheme for each. In the end, we were able to cover about 90% of the H1 HA gene, 86% of the H3 HA gene, and 85% of the BVIC gene. We're really just missing those ends, which is a piece we'll be working on in the future to incorporate those universal primers at the end of the HA genes in the non-coding regions that could be used to really generate that last amplicon we need to get the ends of the virus, but covering the majority of the gene at this point. This is from a preprint showing some contrived experiments with controls. These are whole virus controls accessed through IRR. And we can see here that when we do a serial dilution of H1 or H3, that or in H1, H3, or BVIC in either waste or nucleus-free water on the top, or negative extracted RNA from wastewater on the bottom of wastewater that's negative for influenza, of course. You can see that we got a decent amount of coverage across the HA genes on average at different serial dilutions, showing that we can successfully cover these with controls. In addition to that, we wanted to assess this approach against real-world samples, both clinical and wastewater. And so a little background on Colorado. We have a Sentinel surveillance system across our state for wastewater surveillance. We have 21 sites that cover the 10 all hazard regions in Colorado, so geographically distributed, and then covering some of our tourist locations, like Aspen. We also have 45 additional sites that we can turn on if there's a pathogen of concern that we need to get access to a wider coverage across the state. But for this effort, we chose 187 retrospective samples from Colorado's Sentinel system that were collected between August 2024 and January 2025. We also chose a range of those within 10 log buckets, so 10 to the second, 10 to the third, 10 to the fourth, 10 to the fifth copies per liter, trying to get about 15 to 16 samples of each range that we could assess by these approaches. We also chose over 48 clinical samples that we have through we're both a CDC influenza sequencing center, and so we are able to we're funded to sequence a range of clinical samples throughout the season in Colorado for clinical efforts. And so we took 48 clinical samples we had sequenced during this same time period to check for any dropouts that we could see that were clinically relevant based on the seasonal diversity that was circulating in 2024, 2025. So here we can show that both on the top in clinical and the bottom in wastewater, we have high coverage with our approach in 2024, 2025 samples for H1 and H3. There was a decent amount of both of these viruses, H1N1 and H3N2 circulating during the time period. So that was easy to do. And then we can see that we can cover the majority of the HA gene segment, both in wastewater and in clinical samples. Wastewater, within those, we got high coverage in most of the ranges of copies per liter that we tested, and pretty much down to the sensitivity that we can see with dPCR as well. So we were pretty excited about that. And then in 2025, 26, obviously, we had the emergence of a more divergent H3N2 virus, the subclade K virus that has emerged. And we wanted to assess, obviously, first, whether this approach was still going to be able to pick up and cover or represent this divergent H3N2 in wastewater. So at the time, we took 3 clinical samples that we had at that early in the season in-house, and 6 wastewater samples ranging from, it's actually 10 to the second, so we'll have to update the slide, to 10 to the 4th copies per liter. We can see compared to the serial dilution data we had from wastewater for 2024, 2025, that the clinical subclade in gray and the clinical samples that are subclade K in gray, and the pink wastewater samples that are subclade K heavy or during this time period in pink, we could still get good coverage of the HA gene as well. And so we chose to move forward with performing prospectively influenza H1, H3 sequencing in wastewater during the 2025-2026 season. And Colorado was one of the states that got hit pretty early on in the season. This is a view from CDC's flu view, one of the first weeks where we saw high influenza-like illness activity. Colorado is pretty easy to pick out compared to the other states at this time, and likely due to introduction of, likely due to our tourist sites and high levels of H3N2 at that time. So this is data that's has been submitted and is kind of add on top of the preprint that I've been discussing previously to this. Basically, this is some clinical data from Colorado from this season, from September to February. On the top, you can see these are hospitalizations over time per 100,000 people. And you can see that in B, this is specimens that we've gotten in-house through our clinical surveillance sequencing efforts over the season. You can see those graphs correlate, and that we had a high number of H3N2 subclade K in orange during this season so far in Colorado up to February. And then on the bottom, these are the number of unique wastewater utility sites that were collected by week from September to February that were where we detected flu A in the wastewater. And you can see that the detections correlate well with our clinical data for our wastewater detections by dPCR. In this slide, I'm showing us breaking that down further. Up top, there's the influenza A detections of copies per liter in wastewater, both the line of average and then the individual collection sites are in those gray dots. And then on the bottom, you can see when we were also surveilling for H3 and H1 specific targets in by dPCR and wastewater, that the majority of what we were seeing across Colorado over time was this H3 with some H1N1 being detected at the same time. Here is the prospective or real-time wastewater testing that we were doing with H3 sequencing, the H3N gene sequencing we were performing. On the top, this is percent coverage for a wastewater's collection sample versus the dPCR concentration for flu A over time. And the red dot represents the first sample we got in wastewater. We got over 60% coverage of the HA gene, which was a target we were looking for based on contrived experiments and bioinformatics analysis, as well as it's pretty much the range we've had for SARS-CoV-2 as well. And the dotted line represents when we first confirmed subclade K. Well, actually, it doesn't represent in this. It's what the concentration, well, I'm going to actually say that is something I shouldn't represent in that graph. But in B, you can see that when we're talking about the same data, which is percent coverage of the HA gene versus collection date, we were able to cover most of the HA gene in that red dot earlier than the dotted line, which represents the first clinical sample, which we were able to confirm subclade K in. And so we were able to see subclade K in wastewater about a week before we actually saw this in a clinical sample that came through our surveillance system. This graph represents our retrospective sequencing of the 2024-2025 season. We went back and sequenced every sample we had in-house that had tested positive for flu A or had detectable flu A by dPCR. And this is the average abundance. You can see using Fraiture for different H3N2 viruses from those samples. And the main point being that most of 2024-2025, we had more diversity within the wastewater versus this orange box on the right, which is the beginning of the 2025-2026 season. That is mainly subclade K in that kind of yellow mustard color. And when we went further and sequenced throughout the 2025-2026 season, pretty much every wastewater sample that came in the door, you can see that the majority of what we were seeing in wastewater was H3N2 subclade K and that correlates well with the clinical data we were getting. And obviously this is through Fraiture barcodes that Josh Levy was able to give us at the beginning of the season and provide for that effort. And so really, this effort so far has been to kind of demonstrate the utility of sequencing influenza from wastewater. We are performing similar efforts for H5, and we definitely feel that sequencing the HA gene can generate information on a community level that can help in real time both assess and combine with clinical data for public health response. And so with that, I'll just acknowledge everyone on the team that worked on this effort, including our genomic surveillance program, molecular team, wastewater team, sequencing and team, and bioinformatics team. Molly Hetherington Roth is the primary author on most of this work and performed the diversity study, generated the primer scheme, and performed the bioinformatics and genomic epi analysis on most of this effort. And then on the right, we have our epi colleagues or epidemiologists, Allison Wheeler and Kirsten Weisbeck from a wastewater epi team at Colorado, and Nisha and Deborah from the flu program at our state department. And then obviously CDC for the CDC's National Wastewater Surveillance System for the funding and support on the efforts that Colorado has made in this area. And with that, thank you. These are our, if you want to contact us afterwards, this is our mailing or emailing information, and happy to take any questions.
Thank you so much, Shannon. That was a really interesting talk. There was one question about, this isn't really a question, it was more of a comment about preprints and the links for those. I don't know if it would be easier for them to email you or if you would be willing to put them in the chat, but I will let that participant know that Shannon's email is right here. I'm sure you could get them that way.
We could also send them out to the mailing list, so that might be like a better idea.
Oh yeah, we could also do that as well. Shannon, feel free to send them to one of us, and then we can send those out. Okay, so I encourage everyone to put their questions in the Q&A. We have one here, and I was thinking about this as well. So were you able to detect any avian influenza in these samples, or is it just primarily looking at clinical? And then could this be used to maybe look for novel subclades or rare influenzas that might give us an idea of what might be coming maybe next season?
Yes, so we also have another approach that we're utilizing for H5, and the thing about influenza viruses is they're so divergent that these schemes are really hard to make universal. In clinical samples, you can use the universal non-coding ends of the gene to sequence every flu HA gene that might be present, and it's so very conserved, but in wastewater, because of that fragmented F thing and the fact that HA is like a 1200 base pair gene, it's very hard to get that sensitivity. And so we've had to make an approach specific to H5. We'll probably have to make an approach specific to H7 with tiled amplicon, right? But obviously hybrid capture approaches or metagenomics or agnostic metagenomics would be the way. It would be easier to just detect, identify, and then move from there with that effort. But for characterization and more in-depth information, being able to sequence those specific avian influenza viruses, making a specific scheme against them is important at this point, it seems. And then secondly, we have been able to sequence it in wastewater, and we've done that in real time when we also run our H5 dPCR assay along with the H1 and the H3 in real time. If we ever detect that, then we will perform sequencing for H5 as well. We are working on multiplexing all of these targeted approaches together to make that easier to do in real time, but we haven't been doing that at this moment. We do see the avian influenza when we in Colorado had a very large dairy introduction in 2024. That correlated really well with H5 detections and sequencing in wastewater. We haven't seen a lot of avian influenza otherwise, but that's probably because Colorado's sewer sheds are mainly not combined. And so getting that environmental or avian introduction would need to come from a source like milk, or unless there is, birds that land on a retaining pond in a sewer shed. We have not observed as much of that introduction as I've heard from some other states that have more combined sewer sheds and seem to have heavier migration patterns over those sewer sheds.
Great, thank you. There's also some questions about potentially looking at other gene segments within the influenza. Did you consider looking at either the NA gene? We also have a question here about the M gene, or are you primarily just trying to assess the HA?
We do want to expand out to the other gene segments within influenza. We just wanted to start here for what we felt would be the most impactful piece of information to do proof of concept and understand utility in wastewater. We are expanding out to the NA genes. We have designed an approach against PB2 because of H5N2 and that mutation within the PB2 gene that is concerning for human-to-human transmission within that B3.13, the circulating in dairies. So we have been able to generate but that's pretty specific to what was circulating in 2024 and still to this day, but not as heavily. So working on those pieces, but definitely we've also been able to sequence portions of the M gene when we've been looking to understand if the flu A dPCR assay is still working really well against, that conserved region within the M gene that you would need to pick up all flu A targets. We've seen some diversity there, but that's been more of a really targeted effort of just that region that we pick up in dPCR.
Yeah, there's also another question if you've looked at the Toonie primers. I guess it might be. Have you tried the Toonie primers to get all of the segments? So maybe these are more universal.
Yeah, we've tried approach. We haven't specifically tried the Toonie primers in-house, and basically we want to kind of get our hands on pretty much anything everyone's doing and see what in-house will be the best for, because what we really care about is the public health impact. And if someone else has developed it and we can utilize it in-house and start prospectively assessing the current state in Colorado, we really want to know. We are pretty much constantly looking, constantly cataloging and trying to pull out approaches. And we're going to go retrospectively to some of these samples and compare them to see what we can land on and what we can glean from other groups.
Yeah. And then have you ever are there like methods to look at maybe combining some of these tiled approaches with the more shotgun sequencing approaches so that you could get both the really specific ideas around what H genes are there, but then also more broadly what is there, or is that just not necessarily feasible for what you all are trying to do?
That's something we're looking at for our surveillance system going forward is at least in a subset of samples or in a certain cadence performing targeted amplicon, hybrid capture, and metagenomics to try to be covering all those bases of the pros you can get from all the different approaches at once. And I think that's where a lot of groups are leaning towards in wastewater, is that where the landscape sits right now, utilizing all three of them might be the way to get both sensitive detection, constant expanded detection of anything that's out there, and more characterization of things that are impactful in that moment that you could design more targets or more tiled amplicon approaches to.
Yeah, and then do you have any ideas on how maybe you could distinguish like an H1N1 versus an H1N2? Are you relying more on clinical data? That's obviously a huge problem with these segmented genomes. Do you have any thoughts on maybe ways that you could think about doing that?
I believe that others are heavily thinking about how you might be able to cross-link RNA in the way that you can DNA. So you might be able to sequence peripherally associated H1 or HA and NA genes. I don't know that approach has been really fleshed out at this point. That's the way I could see that working. If you could get some form of single cell sequencing, that would be a way, but obviously this is a caveat to any approach right now.
Yeah, that's always such a challenge when these genes are not physically attached. So without any more questions, it looks like it's 8:58. We are just on time. I want to thank all three of our speakers today for a really interesting and informative new webinar this month. We hope to see you all in May, and I hope everyone has a good rest of their Friday.